Hi, everybody. Before we get started, I'm sure you guys are anxious to start studying for anatomy and everything, but I do kind of want to talk a little bit about how to study during exam week. I'm sure some of you are kind of confused on to how to keep up with the new material and how to study for the exams while you're still getting lectures and all that. So I just wanted to talk about that briefly. So pretty much what I did last year was I would start studying for anatomy or in-depthly studying for anatomy the weekend before. So this past weekend, I would have spent about 60% of the time working on anatomy, and then the other 40% of the time working on the three other classes, and kind of focusing on biochemistry because that's the next test after anatomy. So I would spend most of my time doing that over the weekend. On Monday, so like today, I would focus only on the new lectures of genetics and biochem and kind of link them back to older lectures for biochem and genetics. So I wouldn't do any anatomy on Monday. I wouldn't start studying for anatomy until Tuesday, so tomorrow afternoon, and then I would continue only doing anatomy up until the test. So Tuesday afternoon, evening, whatever, all through Thursday. After that, I would typically go to the lecture after the anatomy test. And then after that, I would go to the TA session for Thursday evening. So in this case, that would be biochem. And then it would be Thursday night through Saturday morning that I would focus on anything but biochemistry. So it would be either genetics or immuno that I would focus most of my time on. And then it isn't until around Saturday, around like noon, until Monday morning that I would continue doing only biochem. And so I would pretty much follow that pattern the following week. So during test week, you will probably be getting new lectures, but most of them will be for the next block of exams. So I would still go to lecture for those just so you don't fall behind, but you don't really have to worry about learning that new material right away. So following the biochem test, I would, work exclusively on genetics, and then following genetics, obviously work exclusively on immuno. So if you guys are still struggling with figuring out how to study for everything and keep up with the lectures, that's kind of how I did it. Hopefully it's helpful to you guys if you need that guidance. So I'm going to go ahead and start going over lectures 6 and 7 now. Kathleen, do you have a question?
Yeah, I just had a quick question. So
Yeah.
the last information for block 1, is that today or is that going to be on Wednesday?
Yeah, so for biochem, your Wednesday information is also going to be on the exam. So
Okay.
that's it. It's just this week's stuff.
Okay, so then is that the same with all the classes?
Honestly, I have no idea. I feel like for the most part, Dr. Muffley, he stops today, so it's a little bit weird. But I think for genetics and immuno, they immuno might have one more lecture on Tuesday, but honestly, I have no idea.
Okay, thank you.
You're welcome. Yeah.
They told us for immuno that ends on the 24th.
Oh, okay. That's convenient. Yeah, so that's this week. So that's awesome. Lucky you guys. I know that sometimes they do have the last bit of lecture material up to 3 days before the exam. So be wary of that. There is a question on the chat. Are we having a practice test on Friday? So I think me and the other two TAs, so not Justin because anatomy is over, we will likely hold a PowerPoint presentation and not actually give you guys a practice exam because you guys will be doing exams all that week. So that would be excessive. But we do kind of want to give you guys some practice questions and probably do it in a PowerPoint kind of presentation thing. We haven't really decided yet, but you will get questions. Any other questions before I start?
Yes. Yeah, so after the anatomy test, you said that we do have the microbiome lecture after that.
Well, no, you would have immuno because it's on Thursday. So yeah, you would have an immuno lecture.
Okay. So that test is it just during the normal block, like the hour 20 minutes?
Yeah, so you would have your test always at 2 o'clock and it would go the hour, well, it would be 70 minutes. So it's a little bit shorter. So you have like a little bit of more gap time between that and your next class. But yeah, you will have a class right after it.
Okay, thank you.
You're welcome. Any other questions? If not, I'm going to go ahead and start. Okay, so I'm going to just go over the 2 lectures that she gave last week. And then on Thursday and Friday, I'll do the this week's lecture. So today's lecture and Wednesday's lecture, and then you'll be all caught up by then. She didn't finish the enzymes lecture for last week, so I'm going to go ahead and finish it for her based on, actually, just kidding, she finished it today. So yeah, we'll just go right through it. Okay, so for this first slide, this is probably kind of just an intro slide, just understanding that membranes are for the purpose of like protecting the inside of the cell, and they also kind of dictate what can go inside of the cell and what can leave the cell. So knowing that these are the 4 things that are make up the membranes is important. I don't know why she indented this, but literally it's just these 4 things. And then you've got your subtypes of carbohydrates. For this slide, don't memorize like what everything is mostly made up of. The main point she wanted to make is that your membranes are going to be either mostly lipid or mostly protein. So certain membranes, like what you would find in the myelin sheath, the myelin sheath that envelops like axons on your neurons, it's mostly going to be lipid. And when you see that something is mostly lipid, its purpose is to protect. Alternatively, on the inner mitochondrial membrane, you're going to see that it's mostly protein, and that's how you'll know that its function is mostly it mostly works as like a functional component, as opposed to like protection. So it's more functional. That's her main point. So here she is just introducing the different types of membrane lipids. You've got your 2 subtypes, your phospholipids and your glycolipids. And then your phospholipids have 2 types below that, and those are your glycerophospholipids and your sphingolipids, specifically that make sphingomyelin. Your glycolipids also make sphingolipids, meaning that they both have the sphingosine backbone, but these make cerebroside types. So make sure you can differentiate between these 2 types of sphingolipids. For this, I think you would be smart to memorize this, because that is your glycerol 3-phosphate, that then when you add the 2 fatty acid chains, which I've highlighted it in blue, this is your glycerol group. So you add the 2 fatty acid chains to the glycerol group via ester bonds, and that's how you make your phosphatidic acid, which is just your general phospholipid without the alcohol group added. So kind of have an idea as to what these reactions are. I would know this word and I would know this word, but like don't really worry about like the actual structure. I would kind of understand the difference between the polar head and the hydrophobic tails. So understand that your hydrophobic tails are going to be consist of glycerol, which is here in green, and then you've got your 2 fatty acid tails. What's important, like super important to know, is that your C1 fatty acid tail, that's this one right here, is going to be saturated, meaning there is no double bond, and therefore it will be pretty straight. It won't kink like your C2, which is going to have this unsaturated bond or double bond, and that's going to lead to this little kinking, and so it allows for the fatty acid tails to not stack, and that helps increase the fluidity of the membrane. So I would also understand that your polar head is made up of this phosphate group here, plus any of the alcohol groups. So this X is just standing for any of these alcohol groups that can be added. Don't worry about the structure for any of these, except I would know that inositol is a ring as opposed to like these other structures. So, but you don't have to know like the structure of the ring or anything, just kind of understand that it is a ring. So this is just another memorization slide. I honestly don't know how important it is, but she did give it its own slide, so I would probably memorize it. So you have to understand that most membranes are going to be made up of lecithin or cephalin. So those are just the common names for phosphatidylcholine for lecithin, and then phosphatidylethanolamine for cephalin. So the difference between these two is their different alcohol groups. Here in red, you'll have a choline for lecithin, and then you'll have an ethanolamine alcohol group for cephalin. So I would know the difference between those two and know that they are found in most membranes. So I would also know that your mitochondrial inner membrane is going to be composed of predominantly cardiolipin. So cardiolipin is different in that its alcohol group is linking two separate phosphatidic acid groups. So you've got your two phosphate groups here, and then you've got your two fatty acid tails with your glycerol. So know that these are like two separate glycerophospholipids that are linked at the alcohol group. And I would also know its like actual name. So she wanted to give you this example of phosphatidylinositol. It is also called PIP when it's, when we talk about it as a second messenger, it's abbreviated as PIP. I don't know if you guys have already learned about this in some of your other classes, but when she goes over second messengers next block, you'll definitely recognize it. I would kind of know its general functions as a second messenger and that it deals with endo and exocytosis and that sort of thing, but I wouldn't really worry too much about it because she's likely not going to ask. What she did want to make a point of is the fact that when you add the phosphate group to these three different carbons, so carbons 3, 4, and 5, you can kind of alter its function a little bit. So these are all examples of this PIP with a phosphate group in different attached to different carbons. And because it's attached to different carbons, it's also found in different regions of the cell and likely has a slightly different function for each of these different compartments that it's found in. But I wouldn't memorize this at all. I just kind of have a general understanding of the fact that it can, be phosphorylated at these carbons, and that dictates whether it's found in one part of the cell versus the other. This you don't have to memorize. She's just saying that if something goes wrong with PIP, you can have all these different types of diseases, but don't memorize it. Okay, so now she wants to start talking about sphingolipids. So this is your other type of glycerophospholipids. So you have your sphingosine backbone, and this is kind of important to know because you're also going to see this in your, your glycolipid backbone as well. So this is going to have your hydrophobic tail, and it's also going to have a polar head that makes it up. It's going to be a C18 chain. So if you count all these carbons, it's going to add up to 18. So that's pretty much what that's saying. I would probably memorize that it's a C18 chain. As for the functions, I wouldn't worry too much about it because almost all of the lipids have very similar functions, and she's not going to ask you one versus the other based on just the functions given. But I would know for the last one that it's what makes up your myelin sheath, hence why it's found in brain and nervous tissue predominantly. Okay, so understanding what makes up a sphingosine and all of this is kind of important, and how you can take the regular backbone of sphingosine that we already talked about, and if you add a fatty acid tail here highlighted in blue through an amide bond, you will now make a ceramide. And now if you take this ceramide, which I would definitely remember because we talk about ceramides a lot, and then you add this phosphorylcholine group highlighted in pink, you now have sphingomyelin. So it's really just building a bunch of things up to make different types of lipids. But as for the actual structure of this, I wouldn't memorize it. I would literally just memorize it as the words. So pretty much what's in this box is good enough. So now she starts talking about carbohydrates. Carbohydrates are really just a bunch of monosaccharides that are linked together into an oligosaccharide chain. So this is just like your glucose, galactose, all these different types of monosaccharides that are linked together to make a carbohydrate. I wouldn't really worry too much about memorizing the different types of monosaccharides because that's not really that important. But I would understand what an oligosaccharide is versus a monosaccharide, which is just one of these. So when you add oligosaccharides to either lipids or proteins, they now become glycolipids and glycoproteins. These will usually be found extracellularly, and they make up, a small percentage of the membrane, but they're also important for stabilizing the membrane because remember, carbohydrates have those OH groups, and those OH groups allow for hydrogen bonding with the water that surrounds the cell, and that's what helps stabilize the membrane structure. Once again, I probably wouldn't worry too much about the function for these specifically because like you'll see like with adhesion and at least cell recognition, you're going to see that with a lot of these different types of like lipids and whatnot. So don't worry too much about it. Okay. So now she's talking about the last group, and this is your glycolipids. So pretty much what the first group is called a cerebroside. So I would know that, like we talked about before, the cerebrosides have a sphingosine backbone, and when you add the fatty acid chain to that, you're now going to call it a ceramide, like we talked about before. So I would understand that when you have a ceramide and you add a glucose or galactose, so only a monosaccharide, to the ceramide, you are now making a cerebroside. Now, when you add glucose, galactose, mannose, any other type of monosaccharide to the glucose or galactose on the cerebroside, you're now making something known as a globoside. So the difference between cerebrosides and globosides is the amount of sugars that you have attached to the ceramide. The next one is the ganglioside. So the ganglioside also has an oligosaccharide chain similar to the globoside. So this is the same. However, you're going to have in this oligosaccharide, please excuse the fact that I connected it that way. It should be the sialic acid is an acidic sugar group. It will likely be found within this oligosaccharide chain. And if you have one or more sialic acids in this oligosaccharide chain, you're likely now going to call it a ganglioside. So understand the differences between these and how to turn one into another, and yeah. But you don't have to worry about the structure, just kind of understand like the vocabulary words. Okay, so she briefly talks about glycoproteins. We kind of already talked about this, as in you have a protein that comes out of your ribosome. After it's made, you have a post-translational modification. In this case, a sugar group or a carbohydrate group is going to be added. We talked about this when we talked about collagen glycosylation of the hydroxylysines. So you should already be somewhat familiar with this, but this is different in that, this is happening on the membrane. This is an awesome slide to review because it kind of just gives you these pictures here. And I think, honestly, these pictures are how I learned the material and how to differentiate between glycerophospholipids and, your sphingolipids and all that. This is really all you need to know in terms of like the structure. For this slide, she's really just saying that most membranes will be mostly composed of glycerophospholipids. And within those glycerophospholipids, you have all your different subclasses based on the type of alcohol group that's attached to it. So she's saying here that the subtype that is most prominent in most membranes is the phosphatidylcholine. You can memorize it if you have extra time, but I wouldn't worry too much about it. Okay, so now she starts talking about proteins. Once again, the function is somewhat similar to all the other membrane components, with the structure and then the receptors and then the transport. That's kind of, somewhat exclusive to it. But regardless, I wouldn't really worry too much about the function because it's mostly just an introduction. I would know the classifications between integral and peripheral proteins, which we'll get into. So integral proteins are going to be found throughout the cell membrane. So they will likely be connected in some way to the cell membrane. Either they go all the way through, or in this case, they are covalently linked to the lipid, phospholipid group. So this would be covalently linked to the phospholipids directly as a lipid anchored subtype of integral membrane proteins, or you have your transmembrane proteins, which is going to be the one that I would say is higher yield. You have to understand that your C terminus is going to be on the cytoplasmic side, and then your N terminus is going to be on your extracellular side. So that's easy to remember because C for cytoplasm, N for not the cytoplasm. And then I would understand that your hydrophobic amino acids are going to be the ones that traverse the cell membrane. So you're not going to find hydrophilic amino acids here because remember, the cell membrane isn't going to like to interact with a hydrophilic amino acid. It's going to be better off with hydrophobic amino acids. However, you can have hydrophilic amino acids on the exterior parts of this transmembrane protein. Remember that when a protein is subjected to water after it comes out of the ribosome, it's going to spontaneously fold in its favorable type of folding. So when it's favorable, it's going to have its hydrophilic amino acids facing the water, and the hydrophobic ones will be hiding in the middle. Okay, so when it comes to peripheral membrane proteins, for these, you're not going to have, you'll have part of the amino acids coming in slightly into the membrane, but the difference is that they're not going all the way through. Your lipid anchors are also a subtype that you'll see. However, they're not going to be connecting directly to the phospholipids like you would see in the integral proteins. These are going to connect directly with things like glycosyl phosphatidylinositol, myristal, and that's really how you differentiate them. I wouldn't worry too much about memorizing these. Or really knowing the difference between integral and peripheral, aside from the fact that, the main difference is being like going through the membrane versus just kind of hanging out on the periphery. Adam, do you have a question?
Yeah, I'm just wondering for lipid anchored, why that is a integral membrane protein versus not being like a peripheral.
I think it's because it's like covalently linking directly to the actual phospholipid versus using one of these like intermediate things to connect to it. Does that make sense?
Yeah, I get that for that side, but then like the one on the left here, that looks to be like directly like in the membrane. So I don't understand how that's peripheral, but
Oh, I see. Yeah, so it would be integral if it's also like entering into the other side, like this, say this is the cytoplasm. If it goes all the way through and then it has like one of these hydrophilic portions on the other side, then it would technically, go through the entire membrane and would be considered integral versus peripheral, which is just going to be on one side of the membrane.
okay, thank you.
You're welcome. Okay, for this, she really just wants you to know that you can have membrane receptors that are either integral or peripheral. It doesn't matter. I wouldn't really worry too much about like what I highlighted here because she's going to talk about signaling pathways in greater detail next block. So just understand, integral and peripheral proteins can be present as membrane receptors. Okay, so this is actually pretty important, and I would definitely star this as an important slide because she likely will ask a question about hydropathy plots. So what these are just ways of trying to predict whether you are going to have a hydrophobic region, so which is likely a transmembrane region of a protein, or your hydrophilic regions, which are likely going to be on the surface of the cell or inside the cytoplasm. So I might not worry so much about memorizing these numbers, but you can if you want to. So your short window sizes are going to be important for finding surface exposed regions. So this would be these areas of the protein. And the reason why you need short window sizes versus large window sizes is because this is going to be a mixture of both hydrophobic and hydrophilic, because it really doesn't matter for it to be just strictly hydrophobic like it would be in the transmembrane region. So because of that, you're going to have to zoom in a lot higher so that you can kind of differentiate it from being transmembrane versus, like the peripheral aspect of it. Alternatively, the hydrophobic regions or the transmembrane regions will all be, hydrophobic, so you don't have to zoom in as much to, be able to differentiate between the two little slots, because these are going to be long sequences of hydrophobic amino acids. So that's why you can use a large window for that. As for these two methods, I remember memorizing the difference between these two, but I don't remember if she actually asked it. So if you have extra time, you can memorize it just in case. And then as for like the actual graph, she's not going to give you the graph, but I would definitely know the values for each of these, whether it's like negative or positive. So if it's positive, it's going to be hydrophobic, and that means this is a transmembrane region of the protein. So this would be all of this here would be a positive value, versus your hydrophilic or negative regions here, which would be like either in your cytoplasm or your extracellular matrix. Don't memorize any of these numbers. I think that's just to look at. So essentially, if you're looking at this region here, you're going to essentially be somewhere within the membrane. So this is your hydrophobic amino acids. You're going to be inside the membrane. And when you pop out under, past this 0 line, you're going to now enter a hydrophilic region, meaning you've just exited the membrane and you're either on the, extracellular matrix side, or you're going to be on the cytoplasmic side. So every time you cross the 0 line, you're changing from inside the membrane to outside the membrane, essentially. Okay, so cholesterol, she talks about briefly. I honestly don't remember her asking a question about it, but I would understand that by adding cholesterol to it, you're going to kind of disrupt the stacking, or the tight packing, as she calls it here, of these phospholipid fatty acid tails. And that allows for, a maintenance of the fluidity so that they can move around still, and that also increases stability. Additionally, by adding a cholesterol, which is hydrophobic, you're going to even more so reduce permeability of hydrophilic molecules, because they're not going to want to go into something that's even more hydrophobic than it would be otherwise. But yeah, I would just kind of memorize these points on this slide. So this is just an introductory slide, kind of giving you the difference between like your just regular fatty acid or phospholipid just by itself. You've got your polar head, your hydrophobic tail, when they all kind of come together into a mycele. This is what would be considered soap. And then when you have 2 layers of them, this is your lipid bilayer, which you would find in your, membranes. As for liposomes, all I could really find about that was that it's more of like a pharmaceutical thing, that they typically put like a polar drug in here, and then, you take the drug and it allows it to enter the cells easier. But I wouldn't worry too much about that. So your fluid mosaic model, Bio 101 vocab word, is going to pretty much tell you what your cell membrane is composed of, and knowing that it's got lipids, proteins, cholesterol is like the main point here. And this is everything that we kind of talked about all put together into one picture. But really, I would just know this term and kind of what it's describing. Okay. So this is talking about your membrane fluidity again. So what she means by fluidity is that these individual phospholipids can move like, in all directions, or they can even flip and switch sides with the phospholipids on the cytoplasmic side, or the, other side of the cell. So I would know kind of the term. So transversal is the switching of sides, and then you've got your lateral diffusion, which is moving from, one side to the other within the same, layer. And I would also know that, having your unsaturated fatty acid tails allows for the double bond to form these little kinks so that they don't stack together so tightly, and that also allows for fluidity. When you add cholesterol, it adds to the stability of it, so it does reduce the flexibility, but it does prevent tight packing, which allows it to maintain a fluid type of structure. So she did mention that temperature has something to do with this as well. We know that kind of like when you think about like olive oil, for example, you know that that's kind of like a liquid at room temperature. So you'd know that it must be above its transition temperature. So if it got any colder, or, ultimately you can make it turn into a solid, but because room temperature is above its transition temperature, it's going to be loosely packed, and that's, it makes it more fluid, and that's more liquidy. Butter, on the other hand, is going to be below its transition temperature, and therefore it is more tightly packed, and that allows it to be more solid. So that's pretty much what she was getting at here, but I wouldn't really worry too much about like the temperature, but knowing these like statements here is kind of important. So for the microenvironments, what she means by this is that because of the fluidity of the membrane and all the stuff on the membrane, so she uses this as an example. This is an integral protein. These groups of phospholipids are all kind of like rigid, and they kind of move together, and they're called a lipid raft. So kind of like a raft on the ocean, they move together, bringing this like integral protein that's hanging out like in the middle of them with it. So as they move together, the protein moves with it, and it could bring it to this other protein, and that allows for like oligomerization and the formation of like a receptor, for example, or like the activation of a receptor. So you put these two proteins together, and now you can activate a receptor so that they can, start binding substrate and activating anything on the cytoplasmic side. So it's important for signal transduction. Okay, so as we know that your membrane is selectively permeable, so it's permeable to small hydrophobic molecules. So your macromolecules, your large molecules, and your charged molecules are not going to be able to cross through the membrane. So she does want you to know, the difference between passive and active transport and the subtypes between those, which I'll go into. So with diffusion, this is going to be only specific to hydrophobic or small hydrophobic molecules, because it doesn't require like any kind of protein or anything. It's really just a hydrophobic molecule being able to go down its electrochemical gradient by moving through the membrane itself. So yeah, knowing that, because it's moving down its electrochemical gradient, it doesn't need ATP or any kind of energy. It's just doing this kind of spontaneously. She had this slide, but she didn't add it to your slides. So she likely won't ask about it because she didn't add it, but I took a screenshot and I added it just in case. So passive transport also has a facilitated carrier protein, and this is for larger molecules or charged molecules that need to enter and exit the cell, but can't do so because of the hydrophobic membrane not allowing it to. So in order to fix this problem, you have these carrier proteins. So your first step is recognition, remembering that your carrier protein is going to be specific for a specific molecule. And so it'll recognize the specific molecule, the molecule will bind to it, it will then transport it across the membrane, release it into the cytoplasm or out of the cell if it's moving in that direction, and then it goes back to its original conformation so that it can recognize the next substrate and so on and so forth. So passive transport is the same kind of thing again. You have these proteins that will allow for charged or large molecules to cross the membrane, like I mentioned. I would know that passive transport has ion channels and gated ion channels, but active transport does not have either of these. What active and passive transport both use, however, is the uniporter, symporter, and antiporter. So these are just carrier proteins that either move one molecule in a certain direction, or a symporter, which moves two molecules in the same direction, or an antiporter, which moves one molecule in one direction and another molecule in another direction. And remember, passive transport is always going to be going in the direction of its electrochemical gradient, so from a high concentration to a low concentration, and the charged, negatively charged molecules are going to be attracted to the positively charged molecules, and that also has a say in which direction it moves. So active transport also uses these three types of carrier proteins. It does not use ion or ligand-gated ion channels like passive transport does, but the main difference here is that it uses ATP because these molecules are going against their electrochemical gradient. Okay, so exocytosis, we know that that's exiting the cell, so you pinch off your one lipid bilayer, like the bottom one that's on the cytoplasmic side. It pinches off, and with whatever molecules are inside, it floats, and then it kind of combines with the top part of the lipid membrane, and then that releases whatever molecules into the extracellular fluid. And then endocytosis, it's the opposite, but the same exact kind of process. She does want to, you don't kind of understand receptor-mediated endocytosis. So specifically, you're going to see this when we learn about cholesterol. But what this is, it's really, you got your receptor here on the membrane. It kind of binds to its substrate, and then both the receptor and the substrate together are taken in by a vesicle, similar to how it's happening here. And that's how it enters the cell. For these, I wouldn't worry about memorizing any of these. She just kind of wanted to give you an idea that these diseases are associated with membrane defects, and then you've got these three different types of membrane defects that can cause these diseases. You're going to learn about these in greater detail as the program kind of goes on, so you don't have to worry about them too much now. And then summary. So if I remember correctly, there is like 1 or 2 questions on the reactive oxygen species lecture, and they were on high yield points. So I'll kind of point out what I think they're going to be, so you guys have a clue as to what to focus on. But essentially, a reactive oxygen species is obviously a molecule that has oxygen in it, but that is also have a highly reactive unpaired electron, and that's what a radical is. I would know these 3 examples because we're going to talk about them a lot. What she's saying here basically is that you make ROS in cells just under normal circumstances. So it's not something that's unusual or bad necessarily. It's just kind of part of your cellular process. So this is a high yield slide. I think she asked a question on either the Fenton or Haber-Weiss reaction, so I would definitely memorize these reactions, if anything. So I did want to mention the difference between a hydroxyl ion and a hydroxyl radical. So this is your hydroxyl radical. Note that your hydroxyl radical only has 7 paired electrons. It's got this one free unpaired electron here. And so that's, what's going to be highly reactive. Hydroxyl ion has the perfect little octet here. It's just got a negative charge. So that's the difference between the two. So this is not bad. This is bad. I would also kind of know that you can make a superoxide from oxygen, and then that can ultimately be converted to hydrogen peroxide, which can then become a hydroxyl radical. So I would know that order of creation of the hydroxyl radical. But you don't really have to know all these little details. That's too much chemistry. For this, I wouldn't worry about this at all. I know she mentioned this enzyme, but that was probably just for your interest. So she wanted to emphasize that in neutrophils and other phagocytes, you're going to have this thing called a respiratory burst, which you've probably already learned about in immuno, but really it's how they kill the pathogens after they phagocytize them. And that also you can have ROS made by like ionizing radiation. So if you're out in the sun, chemicals, drugs, all kinds of other conditions. So there's a lot of things that can cause them. I wouldn't worry too much about memorizing these though. And here's the same thing. It's just kind of telling you that you use ROS in normal cellular processes, and like she lists a few here, but this is just like an introduction to what you're going to learn later on in this course. Okay, so I can't remember, but I feel like this was important, knowing what lipid peroxidation is and how it's, bad. So I would probably kind of understand the steps in order to do this. So you start off with an unsaturated lipid. You have your hydroxyl radical here. When you add the two, you wind up with a lipid radical, which now has an unpaired electron. Following oxidation of that, you will wind up with a lipid peroxyl radical, which can now react with a unsaturated lipid and form either more lipid radicals or a lipid peroxide. I would just kind of know the general steps here, but I wouldn't worry about like the structure or anything like that. And then obviously if you have like this group right here on your lipid peroxide, it's going to totally mess up the membrane fluidity and the structure, and it's not good for your membrane. So this is actually pretty high yield, I would say. Yeah, so I would definitely memorize these enzymes. I think there is a question on it on the exam. This one more likely to be asked about than glutathione peroxidase, because you do have to learn this next block in greater detail. So I feel like she asks about superoxide dismutase, but I could be wrong. So understanding that this enzyme uses these metals, and what it does is it converts your superoxide ion into hydrogen peroxide. And that hydrogen peroxide through the Fenton reaction can then, form this hydroxyl radical. But catalase can come in and convert that hydrogen peroxide into water and oxygen, and can prevent this hydroxyl radical from forming. Glutathione peroxidase kind of works in conjunction with glutathione reductase. However, it's glutathione peroxidase which is taking this peroxide and turning it into water. The way it does that is this glutathione reductase is going to take this hydrogen group from the NADPH, and it's going to break its own disulfide bond. So these two S's are supposed to like indicate a disulfide bond. So when you add those hydrogens, you're going to have your broken disulfide bonds on your cysteines. And then it's going to hand those hydrogens over to glutathione peroxidase to make these two water groups. Yeah. So for the antioxidants, if you have time to memorize like the vitamin C and vitamin E, go for it. I think I did, but honestly, maybe this is more important to memorize, and then maybe polyphenols. But I feel like this is somewhat low yield, so get to it if you have time to do so. Otherwise, the reactions are probably more important to memorize. And then here she's just introducing a bunch of diseases that involve reactive oxygen species. So I honestly wouldn't worry about memorizing these, just kind of having a general understanding as to what kind of disorders are involved with ROS. But don't really worry about memorizing it. And yeah, summary slide. Does anyone have any questions about this lecture specifically?
Hey, maybe for this lecture and just in general, would we not focus on the diseases? Like, we just forget those slides or just like pay less attention to them?
Right, yes. So I wouldn't pay too much attention to them because you're going to learn them in greater detail later on. I think she just wanted to introduce these ideas to you so that when you learn them later on, you can like essentially refer back to this information. But yeah, I wouldn't worry too much about them.
All right, thank you.
You're welcome. Okay, so I'm going to set up the next set of slides. It might take a while because my computer's acting up, but I apologize. So I'm just going to share my screen instead. I'm sorry if like my pointer is not going to be as like obvious, but like for whatever reason, it will never load the second slide. I was having issues with it this morning. So I'll try to like draw as much as I can so you guys know what I'm pointing at. Okay, so pretty much what you have to know here is just like the general, functions of enzymes. You know that they break down large molecules or they build molecules. They help with the making of DNA and they help cells communicate with each other. It might be also important to understand that when you inherit genetic disorders, many of them are due to a deficiency or a lack of enzymes. And so if you don't have the enzymes that perform the proper function, you won't be able to do that function in the cell. And that's, the cause of many types of diseases. Additionally, you can actually have an increase in the activity of these enzymes, such as enzymes responsible for cell replication. So if you have an increase in these enzymes, they'll increase the cell replication, and that can lead to cancer. She'll talk about how enzymes are kind of looked at when you draw blood from a patient, and it can help you determine the function of their tissues or organs based on the amount or activity of these enzymes. And then, she talks about prescription drugs, how many of them, and you'll learn about this in greater detail in pharmacology next semester, how these prescription drugs are useful for activating or inactivating or altering enzymes, and that's how they, affect the system. She said not to memorize this because you will have to learn this next block, but kind of understand that if you have a defective enzyme, such as phenylalanine hydroxylase, you are unable to, do a particular reaction, and that's not good. Okay, so she does want to emphasize that your enzymes are going to be highly specific for its particular substrate. They are also going to increase the rate of the chemical reactions, but they're not going to change how much product or how much substrate is being used or made. They're really just affecting the kinetics of it. If you guys remember from when we talked about collagen or hemoglobin, most of your proteins that are enzymes are going to be globular proteins. And this is important because the globular proteins, remember, have a dynamic role, so they kind of can be altered in terms of their shape. They can change conformation, and remember that that's important for, when something binds to the active site, for example. You can also have one subunit or multiple subunits, like in hemoglobin, which has 4 subunits, and we saw that as an example a few lectures ago. Don't worry about memorizing any of these reactions. But the point she wanted to make is that these reactions happen by themselves. You don't have to do anything to them. So because they are spontaneous reactions, they are able to be catalyzed by an enzyme. So you can increase the rate of these reactions by adding an enzyme, but the enzyme by itself will not take a reaction that would not happen spontaneously and make it occur. It would just simply increase the rate of the reaction. So understand that enzymes do not affect the thermodynamics, they only affect the kinetics of a reaction. So for this, I would probably memorize that OMP decarboxylase has the greatest rate of enhancement of all the enzymes that we know of. And you can kind of see that here as being like the highest number compared to all of these. So the rate of enhancement is really the reaction rate of a reaction with the enzyme over the reaction rate of the reaction without an enzyme. So that can tell you how much better the reaction occurs with the addition of an enzyme. Don't memorize these numbers at all. That's too much information, but kind of just have an idea that, you can increase the rate of the reaction by adding an enzyme. Here she's just introducing the active site and the substrate. So your active site is going to be the location on the actual enzyme that interacts with the substrate being the reactant, essentially. Okay, so these are 4 things that you have to know about enzymes. Like I said earlier, the enzymes aren't going to allow a reaction to occur that wouldn't occur on its own. So if you have a reaction that it has a positive delta G value, remember that's, a non-spontaneous reaction, the enzyme is not going to allow it to happen. You would have to, input energy for that to occur. But yes, so therefore it must have a negative delta G for the enzyme to be able to work with it. The second rule is that, your enzymes are not going to be permanently altered. They will change their conformation when you bind the substrate. But when you release the substrate, it's going to go back to its original conformation. Also, it's important to know that, the reaction can occur in the forward reaction or the reverse reaction. So if you have a ton of substrate, it can bind to the enzyme and then form products. But, if you have a lot of products bouncing around and they, enter the enzyme can convert it back to the substrate. So really it, can go in either directions, and it goes in one direction more favorably based on the amount of substrate or product available. And lastly, enzymes are highly specific for the substrates, meaning that only one specific substrate is going to bind to that specific enzyme, and therefore it can only do one reaction at a time. Okay, so like I said, enzymes do not affect the thermodynamics of it, and therefore will not affect the equilibrium. So the equilibrium constant will not change, only the activation energy. So you have your free energy of, your substrate, and this is the free energy of the products. As you can see, your substrate is at a higher energy level than your products, so you know that this is going to be an exergonic reaction. It's going to release energy when it goes to the products. So the enzyme will not mess with the free energy of the substrates, and it's not going to mess with the free energy of the products. It's only going to mess with this free energy associated with the transition state. So we know that the transition state is going to be at a really high free energy, and that's why it's so unstable and only lasts for like a split second before it turns into the product. So by allowing this, by lowering the activation energy needed to reach the transition state, your enzyme can now much easier and quicker form the product. So really what you have to know here is that your enzymes are not going to change the free energy of the substrate or the products, only the free energy of the transition state. And here she is just kind of reiterating the same kind of concept. You have your uncatalyzed free energy of the transition state, which is up here, versus the catalyzed free energy of the transition state, which is significantly lower. So here you have another exergonic reaction where the substrate's free energy is greater than the product's free energy. So that's not going to be altered, but this activation energy is going to be altered. Yeah, I think that's it. And then also knowing that, you don't affect the equilibrium. So your equilibrium, so the amount of substrate and product that you have at equilibrium is not going to change. But you can reach equilibrium faster because of an enzyme. So that's the important difference. Okay, so definitely memorize these four, specific things about the active site. So the proximity effect is really just essentially being able to bring two reactants together. So you have two substrates that would otherwise need to somehow kind of bump into each other or get close to each other to react. But with an enzyme, the enzyme kind of pulls them both in so that they can fit perfectly in here and are more likely to react since they're closer to each other inside the active site. The orientation effect is kind of the same thing, but because of the shape of the active site is so specific for this reaction, it's going to draw in each of these substrates in the perfect direction so that their functional groups that need to function or react with each other are going to be right next to each other. So it works perfectly for them to kind of instigate a reaction to occur. The promotion of acid-base reactions has to do more with the amino acids found within the active site. So if you have an active site with a bunch of basic amino acids, those are going to be positively charged, and they can kind of give hydrogen ions to the substrates in order to kind of promote a reaction as well. And lastly, the exclusion of water. So if you have two polar substrates, they're going to be interacting with water if they're, just not within the enzyme. So the water is going to be almost like a distraction to these two substrates, and that keeps the two substrates from reacting to each other because they're being distracted by the water. So when you remove the water, you're kind of making it so that they're more likely to react with each other. Okay, so know these vocabulary words, know the difference between lock and key and the induced fit model. So lock and key is essentially saying that the active site of the enzyme is complementary to the substrate. So the substrate will fit perfectly into the enzyme, and that we have decided is not the actual thing that happens because it just stabilizes the substrate and makes it less likely to form product. The one that we have accepted to be true is the induced fit model. This is where the substrate is going to bind to something where it kind of fits in okay, but the shape of the enzyme is going to be more complementary to the transition state. So it will force the substrate to kind of turn into the transition state, and when it does that, it allows it to form product faster and easier. So same thing here. You see that without an enzyme, you're going to have your metal stick, and you're going to have to put in a lot of energy to bend the stick. So the bent stick is the transition state that ultimately breaks it into two pieces, which is the product. So this is how much activation energy is required to reach the transition state up here. With the lock and key model, you're also going to have an increase in activation energy because now you're sticking the substrate in a enzyme that fits it perfectly, and that's not going to promote it to bend or break, and so you're not going to be likely to form product if it's just stabilizing the substrate. So that's why your energy of activation will increase with if we're following the lock and key models, hence why that's wrong. Okay, so this is showing you the induced fit model. So you have a active site that is complementary to the transition state. So it's got more of a V shape. If you stick the stick into the active site, these magnets are going to pull it up so that it's forms a transition state easier, and it also stabilizes the transition state so that you lower the activation energy when you form this transition state, and that allows for the decrease in activation energy and the formation of product easier. Okay, so same thing here. You've got your mechanisms. You've got your proximity effect shown here. You're putting the two substrates next to each other, and you're orienting them in such a way that their two functional groups are going to be more likely to react with one another. You're also doing the promotion of acid-base reactions. You see here that you've got a negative and positive charge on the actual enzyme. So this is kind of like having two functional groups or two acids or an acid and a base on the actual enzyme, and that's kind of promoting the acid-base reaction for the substrate, which allows it to form the product or react easier. And those four mechanisms will allow for an increase in the strain that causes the substrate to form the transition state and ultimately form the product. So this is talking about how the binding energy specifically helps kind of lower the activation energy. So your binding energy is going to be like these hydrogen bonds between the substrate here and the enzyme. So you've got your enzyme side chains here. So that's how the substrate is going to be specific to a particular enzyme because it's going to be complementary to the side chains on the enzyme. And so it can form like hydrogen bonds in the correct locations, and that's how you kind of draw the substrate into this particular enzyme. And by the formation of these weak interactions or hydrogen bonds, you're going to be releasing free energy, and the free energy is actually going to go towards the energy of activation. So you're releasing energy and you're putting it back in to the energy of activation required to form the transition state, so that even more so lowers the activation energy and makes it more likely to form product. So kind of understanding that your amino acid side chains in the protein are going to be specific for the substrate that it is going to want to draw in. So we're kind of going over that again, but more specifically for serine proteases. So you guys remember serine proteases as being trypsin and chymotrypsin. We talked about them in like one of the previous lectures. But remember that trypsin is specific for lysine and arginine. So its active site is going to likely be negatively charged. Why? Because lysine and arginine are both positively charged amino acid side chains. So it's going to draw these in and likely bind right here. And then it's also going to be having these serine groups here that will interact with this site right here and allow for hydrolysis. So now you have chymotrypsin, and it kind of does the same thing. However, it's going to have a different looking active site because it's going to be specific for aromatic amino acids. So remember that aromatic amino acids are composed of bulky hydrophobic side chains. So as you can imagine, you need to make sure that the active site has tiny side hydrophobic amino acid side chains. So you're going to have a lot of glycines. Remember, glycines are going to have just a hydrogen as their side chain group. So that leaves plenty of room for the bulky side chains to fit. And that's kind of how you make the active site specific for these types of amino acids. And so again, you're going to have serine groups as well in the active site, but these are going to actually act on this bond here and allow for hydrolysis. So you've got kind of two things going on in the active site. You've got the specific amino acids that kind of draw it in, and then you've got your serine amino acids that kind of actually perform the action of breaking the bond. So same thing here. She's showing you that chymotrypsin's active site. So they kind of look like little test tubes, but these active sites you see for chymotrypsin are all going to be composed of small side chain amino acids. So you have plenty of room to fit the aromatic amino acids. For trypsin, remember that arginine and lysine are positively charged, so it's going to want to have negatively charged amino acid side chains to kind of draw in the positively charged side chains of arginine and lysine. And then elastase, I don't remember what this is specific for, but we don't have to know it. It's also got small hydrophobic amino acids that are drawn into the side chain. Okay, what you do need to memorize is the catalytic triad. So the catalytic triad are the three side chains that are found, or the three types of amino acids that form the functional group of these serine proteases. Remember, serine proteases are your trypsin and your chymotrypsin. So what happens here is that your aspartate has this negatively charged functional group, and it's going to go and grab the proton of the histidine. That double bond can move over, and that gives this nitrogen here a negative charge. So it's going to go and grab the proton from the serine. At this point, now you've formed an alkoxide ion. So maybe know that an alkoxide ion is formed, but don't worry too much about how this is formed. Just know alkoxide ion is the functional aspect of the serine protease that allows for hydrolysis of the peptide bond. And then obviously know that these three amino acids are involved. Okay, so for the serine proteases, you obviously have to know the three amino acids that make it up, but you do have to know that these three other types of proteases exist. Don't worry about like the glutamine or anything. Know that zinc metalloprotease has a zinc aspartic protease has an aspartate, and then cysteine obviously has a cis. But don't worry about these other, side chains. Oops. Okay. For this, she's just kind of showing you that, when you add the substrate to the to the enzyme, you're going to form a conformational change that is kind of triggered by the weak interaction. So like the hydrogen bonding of the substrate and the enzyme. So those form those weak interactions that kind of change the conformation of the enzyme. And then as soon as the product is formed, the the substrate is released and the enzyme goes back to its original conformation. She's just kind of showing you an example of this, and she's introducing an example of a cofactor, but don't worry about memorizing this reaction at all. Same kind of thing here. You have your substrate binding, you have those weak interactions as indicated by these little dotted lines, and that allows for this to move in this direction to form more weak interactions with the substrate. The substrate is turned into the product, it is released, and you go back to your original conformation here. Once again, you're not going to have to memorize this yet. You'll memorize it next block, so don't worry about it. Okay, so knowing the difference between an holoenzyme and an apoenzyme is, I think, a question that she asked on the test, if I remember correctly. So definitely know this. So know that your apoenzyme is just going to be like the protein aspect of it. And some enzymes will also have cofactors. So this is going to be, does someone have a question? Okay. Catherine, did you have a question?
Yeah, I had a question about like a few slides ago with your practice questions. Basically for when we're like looking at the like the serine proteases, should we just think about like what's opposite to them? So acidic goes with basic amino acid, and like bulky like phenylalanine or tryptophan goes with the smaller one, and like vice versa. If she were to ask like, oh, if there's like a smaller one to go with like the larger one, I don't know if that makes sense.
Yeah, no, I get what you're asking. Yes, so yes, your acidic would want to be with basic. So your acidic substrate would be attracted to a active site with mostly basic side chains. The only thing I would know that's not the opposite is that hydrophobic likes hydrophobic and hydrophilic likes hydrophilic. So those aren't opposite, but like small side chains will go with big substrates and vice versa. So yeah. Does that make sense, Catherine?
Yeah, thank you.
You're welcome. Okay. So you've got your apoenzyme, which is just the protein part. So if you think about hemoglobin, remember that it's got the cofactor, which is just the heme group. So that is the non-protein part of the enzyme. And your apoenzyme is going to be the all-protein part of the enzyme. So when you add these together, they make the holoenzyme. So know that the holoenzyme is composed of these two. So the way I remember that is because the holoenzyme is the whole thing. So apoenzyme, cofactor. And then your cofactors can come in two different types. You've got your metal ions. These are just some examples. Don't worry too much about memorizing them because there's so many different types. Or your organic or metallo-organic molecules. And these specifically are also called coenzymes. An example of a metallo-organic molecule that we've already learned about is the heme group. So that is going to be a metal with an organic molecule, so just a bunch of carbons and hydrogens, your porphyrin rings, I think, would be organic molecules by themselves if they didn't have the iron. So knowing that a coenzyme is a type of cofactor, so a coenzyme is always a type of cofactor, but a cofactor is not necessarily a coenzyme. So know that cofactor is the umbrella term, coenzyme is a type of cofactor. And then don't memorize these, but she was giving you other examples of cofactors. Same thing here. Just kind of understand that coenzymes are made by vitamins, so that's why it's important to take your vitamins. So I'm sure you guys are kind of familiar with some of these cofactors or coenzymes, and they actually use like different type of vitamin rings in order to build themselves. So don't worry about memorizing these examples, but just kind of have a general understanding that vitamins help create coenzymes. Okay, this is high yield. I would definitely know how enzyme nomenclature works. So know that your nomenclature is made up of 4 different numbers. The only one that you will have to really know about is going to be your first class. So your first number, which is your class. Your class is also telling you what kind of reaction the enzyme does. So your class number 1, if this number was 1, it would be an oxidoreductase. So this is your oxidation and reduction reactions that we've talked about. I would remember this is the first one because 1 starts with O and oxidoreductase starts with O. Same thing here. 2 is T for transferase. And this is where you transfer a group from one substrate to another substrate. Number 3 is your hydrolases. So this is just your hydrolysis reactions, which involve water. And I remember this because water has 3 atoms, so it's number 3. I don't have a fancy way of remembering these last, but I'm sure you guys can think of something. So lyases is just the formation of double bonds. Isomerases are like transferases, but they're a transfer of a group within the same molecule, so not between 2 different molecules. And then you've got your ligases, which is putting 2 molecules together using ATP. And don't memorize the examples for those. Same thing here, don't remember the subclasses. I thought this was super helpful for being able to kind of understand what's going on for these different reactions. So if you're like kind of stuck, I would kind of look at these pictures because they're kind of neat. But otherwise, don't really worry about it. So here, she's never going to like tell you to guess what this is or this is or this is, but you should understand what the second, third, and fourth numbers stand for. So understanding that the second number is going to define the substrate functional group, the third number will define the acceptor molecule for the reaction, and then the fourth number will kind of tell you the order in which it was discovered. The only ones you would have to know are, the first number, which is the class type. So, yeah, if she showed you this reaction, which she won't because she's not going to tell you to kind of guess what kind of class this is, at most she'll give you this and she'll tell you what class is it, and you would have to know that it's a transferase. But everything else I wouldn't worry about. Okay, so this is really just showing you how you have 2 substrates that enter this enzyme. And remember, when they enter the enzyme, you'll have these like weak interactions. So not hydrophobic, your hydrogen bonding. And that hydrogen bonding is going to release this binding energy that is going to go towards the activation energy and helps lower it even more. This lowering of the activation energy will speed up the reaction, and ultimately your substrates will be converted into products. They're kicked out, and the enzyme goes back to its original conformation so that 2 new substrates can enter again. So it's kind of, an introductory slide as well on the idea of like regulation of the pathways. So usually a product is going to go back and go upstream to down regulate an enzyme. And this enzyme is typically going to be your rate limiting step or your irreversible step. So your irreversible step is the same thing as your commitment step, which we talked about in the purine and pyrimidine pathway. So kind of know those terms as being interchangeably. You can use them interchangeably. And then you've got the other option of having a downstream or, in a different pathway, you have this product that can also come back in and upregulate or even downregulate other processes. So it's really just this introduction of feedback inhibition or regulation, and I wouldn't worry too much about it. At most, know that it's the rate limiting step or irreversible step that is usually regulated by products. So this is her introducing the idea of proenzymes. Proenzymes are going to be important for, I think, Wednesday's lecture when we go over like the blood clotting cascade. So essentially just knowing what this is and how it's activated would be enough. I wouldn't worry about memorizing these examples because this is physiology and that's just too much. So essentially you start off with this whole polypeptide chain. It's going to be inactive because it's got this pro sequence, it's got this activation site, and then it's got the actual active sequence that works. So your regulatory protease is going to bind to this activation site right here, and it's going to cleave the enzyme into a pro sequence that eventually gets degraded, and your active enzyme. So this active enzyme is now activated in order to function. It's important to know that this is also an irreversible process, meaning that the proenzyme cannot return to its inactive form once it's broken. It's activated, and it's activated until it's degraded. Yeah. So isozymes, she was kind of telling you about this. It's these enzymes that have a different amino acid sequence. So an example of this is LDH, lactate dehydrogenase. So lactate dehydrogenase is found in almost every tissue in the body, every type of cell, and it's going to be found as isozymes. So it's going to have the same function, it's going to perform the same type of reaction, but it's going to have a different amino acid sequence, which she said can affect the physical properties of the enzyme itself. So you'll find one type of lactate dehydrogenase in, say, your heart cells and a different type in your liver cells. So they're specific to the tissue that they're in. They play regulatory roles, and then as she went in detail about is that they can be used as biomarkers. So she showed you these pictures here about different time after a heart attack. So right after a heart attack, you're going to have type 2 LDH being the most elevated, but as time goes on, you'll start to see an increase in type 1 being the most elevated. And if a doctor draws your blood after a heart attack, he can pretty much determine, based on analyzing these isozymes, how long ago it was that you had the heart attack. And she shows you this here too, but like with different types of enzymes, so not of the same type. She shows you that you can also measure different types of enzymes, how quickly or how much you have of each to determine, how long ago you had a symptom. And it's also used to determine if you have something that's not functioning correctly or some kind of tissue damage, organ damage, etc. But I think I memorized these like different enzymes, but I don't think she asked about it. I think it's enough to understand that measuring enzyme concentrations in the blood can help you determine the cause of a disease or determine how long ago you had a heart attack, for example. And don't memorize these. It's just kind of the same idea here. You have different enzymes found in different tissues, and by measuring these enzymes, you can determine, for example, your heart or liver function and all of that. And then this is a summary slide. This is a summary slide. And yes, does anyone have any questions about that? I did make a Kahoot, so if nobody has any questions, we can jump to that.
Hey, Phoebe.
Yes.
I have a question. I meant to ask this earlier. Sorry about this.
It's okay.
Would you be able to go back to the slide that's titled Binding Energy and Other Factors Contributing to Reaction Specificity in Catalysis? It's a little bit higher up.
Yeah, let me look for it. This one?
Yeah, could you just review this slide again one more time?
Yeah, so, okay, so you have your protein, and these proteins have different like amino acids in the active site. So for example, in this particular example, you have this substrate that is uracil, and it's going to have these, its active site, or the enzyme that it's specific for, it's going to have active sites that have this threonine side chain and this serine side chain right in the perfect location for uracil to bind properly. So that's kind of how it determines like specificity for the substrate, by having the correct amino acid chains in the perfect order in order to bind with the substrate. So when you make these weak interactions, so these hydrogen bonds, for example, you're going to release free energy, and this free energy is going to kind of be added into the activation energy necessary to form the transition state. So you're taking the energy from this weak binding and using it towards reaching the activation energy required to form the transition state. And I think that's like the main point of this slide.
So when the substrate binds to the active site, it's a weak binding that releases energy, and then that energy drives the reaction to transition state?
Yes, it goes like towards it. I mean, I don't know if it does the whole work, but it goes
But it
towards like.
to it, yeah.
Yeah.
Okay, perfect. Thank you so much.
You're welcome. Anyone else have any questions? Oh, Kevin, a more stable transition state means a lower activation energy, and an unstable transition state means a higher activate. Yes, correct. You make it more stable, you lower the activation energy. Any word of advice for studying the purine and pyrimidine metabolism? Been writing the steps and creating Quizlet so far. Anything unique to try? I didn't try this, but I know some people last year, they made up a song to remember it. I feel like that's like really hard to do, but if it works for you, I mean, it's worth trying. I've also, like, personally for pharmacology, not so much for like memorizing pathways, but you can like kind of make stories about like the pathways and have like link each enzyme and product and substrate to like a story, and as you go through the story you kind of like remember each step. I don't know, that's probably more better for pharmacology and remembering like drugs and stuff, but hey, it might work. Any other questions?
Yeah, I have a question. Could you go over the isoenzymes again, please?
Yes. Okay. So isoenzymes are really just the same kind of enzyme. So it does the same, it catalyzes the same type of reaction. So for example, you have LDH, so you have LDH type 1, LDH type 2, and say these different LDHs are going to be found in different tissues. So maybe LDH type 1 is going to be found in your liver, and LDH type 2 may be found in your heart cells. So you can kind of determine based on the type of LDH, what tissue it's coming from. If it's elevated LDH type 2, you can hypothetically determine like, oh, LDH type 2 is found in my heart. It must be a heart issue, and stuff like that. Additionally, I would know that you can measure these isoenzymes during different stages of specifically like a heart attack. To determine how long ago it's been since you've had a heart attack, because at different stages of the healing process following the heart attack, or the more damaging process of the heart attack, you're going to have different elevations of these different types of isoenzymes. So just knowing that it's the same type of reaction that they catalyze, but they're found in different tissues, and they have different amino acid sequences that can alter their individual physical properties. Does anyone else have a question? Catherine, do you have another question, or? Okay, so if nobody has any more questions, I can start the Kahoot. Okay, I think I'm gonna start it now. It looks like everybody's in. Cool, yes, you would definitely have to know that the C1 is saturated and the C2 is unsaturated. But looks like most of you got that right. Awesome. Nice. Yes, hydrophobic portions will be positive. You don't need to know the graph for this, but I would know which ones are positive, which ones are negative in case she asks. Nice. Yes, ceramide. Remember, all of these will have the sphingosine backbone, and then when you add the fatty acid tail to the sphingosine, it's known as a ceramide. So yes, all of these have ceramide. Nice job. Great, yes. Remember, gated ion channels and like just regular ion channels are only found in passive transport. Active transport and passive have all these other types of uniporters, symporters, and antiporters. Okay, yes, a little tricky. Definitely know this reaction because she's probably going to either ask about that one or the Fenton reaction. So remember, Haber-Weiss, W for water, so you know you're going to have water as a product in this reaction. Yeah, so that's how I remember it. Nice. Yes, cysteine is not part of the catalytic triad. Only aspartate, histidine, and serine are. Cool. Yes. Class 2, 2 for transferase, T, T. They both start with T. Remember it that way. But nice job. Right, yes, it is zinc metalloprotease, not iron metaboloprotease. Nice job, guys. Nice. Yes, you are going to be promoting acid-base reactions, and you're also doing the exclusion of water. You're not reducing the free energy of the products. Remember, that's a thermodynamic property, and enzymes don't do that. And then you're not going to have a complementary active site to the substrate. You're going to be complementary to the transition state. Right, yes. Remember, holoenzyme is going to be made up of an apoenzyme and a cofactor, so it's the opposite. Nice job. Nice job, holoenzyme, whoever you are. Okay. I can stay for any questions you may have about like studying, the material, the tests. But you guys are free to go if you have no questions.
Hi, Phoebe. I was wondering if you could go over slide 18 again, just the catalytic triad, the like specificity of like the binding.
Yeah. Okay. Like this slide?
Yeah.
Okay, so you don't really have to know like the reaction that it does, but knowing like the three amino acids that make up the triad is important. And then, I mean, knowing the alkoxide ion is what actually is like the functional aspect of it. But if you want me to go over like the whole, I guess, the organic chemistry of it, I can. So essentially you have this aspartate that has this negative charge at pH, your normal body's pH. And because of this negative charge, it's going to kind of go and grab this proton from the histidine. Because of that, now you're going to have this like double bond that kind of moves and, you know, on this side and this side, but it can give this one a transient negative charge and allow it to grab the proton from the serine. And by grabbing the proton, now you have this negatively charged alkoxide ion, and that alkoxide ion is going to be the functional aspect of the enzyme, allowing it to like hydrolyze the peptide bond. Yeah, so that's pretty much how these serine proteases work. So like trypsin, chymotrypsin, but I wouldn't really worry too much about like the actual like, you know, what takes what, and, you know, the structure or anything like that. Just memorizing the catalytic triad. That is found in serine proteases, and kind of knowing that the alkoxide ion that is formed from the catalytic triad is what actually hydrolyzes the peptide bond. Did that answer any questions you may have?
Yeah, that was perfect. Thank you.
You're welcome. Any other questions? You're welcome, guys. Good luck on anatomy on Thursday. I know that's like super stressful, but if I remember correctly, the first test wasn't terrible. It was just a lot of material. So good luck, guys. You're welcome.